Hello student, all of you. You are most welcome on my channel, that is Liberty Solution. In my previous video, I have discussed about chapter number 10 of class 11th NCERT biology, that cell and cell division. And here in this video, I will be discussing about chapter number 13, photosynthesis in higher plants. If you have not subscribed my channel, subscribe it. These videos are useful for your school examination, board examination, as well as NEET examination. So let us start today's class that is related with photosynthesis in higher plants. Like, comment, share it with your friends, and don't forget to subscribe my channel. So what is photosynthesis? First, we'll be learning what is photosynthesis. Now, photosynthesis is a physico-chemical process. Sometime question will be there. Photosynthesis is which type of process? So it is physico-chemical process. Sometime it will be in option. It is a physical process. It is a chemical process. So it is what? Physico-chemical process, because there is an involvement of physical energy, that is what? Light energy. Then there is an involvement of some chemicals. That's why it is called what? Physico-chemical process, by which green plants use light energy, that is solar energy, to synthesize organic compound. What is the product? Organic or inorganic? So option will be what? Organic compound. There is a production of organic compound. It is the basis of life on earth. So for what is the basis of life on earth? That is what? Photosynthesis. Ultimately, all living forms depend on sunlight for the energy. So all living organisms, directly or indirectly, they are dependent on photosynthesis, and that photosynthesis is dependent on light energy. Therefore, all living beings are dependent on sunlight for the energy. Now, what is the importance of photosynthesis? It is the primary source of all food on earth. It releases oxygen into the atmosphere. So it is what? Photosynthesis is having major importance, or what? It is the primary source of food on earth. And during this, oxygen is released. And therefore, it is photosynthesis is also source for oxygen in the atmosphere. Now, experiment related with photosynthesis. There were some experiments conducted with related with photosynthesis. First experiment, variegated leaf experiment. Variegated leaf experiment. What this experiment talk about? Suppose this is a leaf. This is what? Variegated leaf. This is what? A variegated leaf. Some area is green and some is yellow area. Why it has become yellow? Because it was covered with what? Black paper. So this is what? Variegated leaf before starch test. Now take a variegated leaf. How will get variegated leaf? Take the normal leaf and some area covered with black paper. So if the area is covered with black paper, that area will turn yellow in color. Means that area will not perform photosynthesis. So the leaf will become what? Variegated leaf. So take a variegated leaf, a leaf partially covered with black paper. What do you mean by variegated leaf? Leaf partially covered with black paper that was exposed to light. Test the leaf for starch. Then you have to do the test of starch. It shows that photosynthesis occurs only in green part. This is the green part. This area will give the starch test, and this area, which is yellow in color, it will not give what? Starch test. Why? Because this green area will perform photosynthesis. So if it performing photosynthesis, product of photosynthesis is starch. So this area will be giving a starch test. This yellow area, it will not perform photosynthesis. So no photosynthesis, no production of starch, therefore no starch test. So it shows that photosynthesis occur only in green parts of the leaves in presence of light. So these are the area unaffected by starch test. These are what? Colorless. Why? Because there is no starch. An area affected by starch test, that is, it turns blue black. So wherever there is a what? Starch is there. If you are doing starch test, that area will become blue black color. So this is a variegated leaf before starch test. This is variegated leaf after the starch test. Now second, half leaf experiment. Second experiment is half leaf experiment. This is a plant. A part of leaf is enclosed in a test tube containing KOS soaked cotton, which absorb carbon dioxide. So this is a plant. Plant is having leaf. Half leaf is covered in a test tube containing KOH. Here there is a KOH is there. KOH means potassium hydroxide. So here cotton is there. That cotton is soaked with KOH solution. What is the why it is soaked with KOH? Because it absorbs carbon dioxide. So whatever carbon dioxide is there in this test tube, okay, it will be absorbed. The other half of leaf is exposed to area. This area will be exposed to air. Now you have to place this setup in the light for some time. Now test the leaf for the presence of starch. So you have to keep this for some time in the sunlight, and then after some time you have to remove this leaf and do the starch test. Now that area which was enclosed in the test tube, that area will not give starch test. Why? Because it was not exposed to sunlight, it was not exposed to environment. So there, so exposed part shows positive for starch and portion on the test tube shows negative. This proves that carbon dioxide is required for photosynthesis. So in the environment, there is a carbon dioxide is there here, but here inside this, there is no carbon dioxide. Whatever carbon dioxide was there, it was absorbed by KOS. So no carbon dioxide, no starch. So in first, we have done that if the area is covered variegatedly, light is required. Here it shows that carbon dioxide is required for photosynthesis. Now, next one, next experiment was done by Joseph Priestley. Experiment by Joseph Priestley. This is the experiment done by Joseph Priestley in the year 1977. He placed a mint plant in the bell jar. Mint, this is a mint plant. He kept this mint plant in a bell jar. This apparatus is called what? Bell jar. He found that the mouse stayed alive and the candle continued to burn. So mice was alive and the candle was okay. The experiment by Joseph Priestley, this was done, he have done the experiment in the year 1770. Priestley performed experiment to prove the role of air in the growth of green plant. Air is required for the growth of green plant. He discovered oxygen in the year 1774. So who discovered oxygen? So the answer will be what? Joseph Priestley. In which year? 1774. He observed that a candle burning in a closed bell jar get extinguished. Similarly, a mouse suffocated in a closed jar. This is a candle, this is a mouse. After some time, the candle is what? Extinguished and the mouse suffocated. He concluded that a burning candle or a breathing animal damaged the air. What this experiment shows that this burning candle as well as breathing animal, they damage the air. In second step, he placed mint plant in the bell jar. This is a bell jar where he have kept mint plant. He found that mouse stayed alive and the candle continued to burn. Why? Here there was one thing was missing. Mint plant was missing here. No mint plant. No mint plant. So the mouse died and candle extinguished. Here only one, there is a difference in setup. This mint plant. Mint plant is there, even candle is burning. Mouse is also alive. Why? Because during photosynthesis, plant release oxygen. So whatever oxygen is consumed, that is being replaced by this plant. So candle is also not getting extinguished and mouse is also not dying. He hypothesized that plant restored to the air whatever breathing animals and burning candle removes. So plant have restored air. Air here, oxygen. Then there was a experiment by John Ingenhousz. John Ingenhousz. The experiment was conducted between 1730 to 1799. He conducted the same experiment by placing it once in the dark and once in the light. He showed that sunlight is essential to the plant for purifying the air. So this is the setup. Once he kept this setup in the sunlight and once he kept this setup in the away from the light. So he showed that sunlight is essential to plant for purifying the air. He repeated this experiment with an aquatic plant. Aquatic plant means the plant which grow in water. They are called aquatic plant. It showed that bright sunlight, small bubbles were formed around green parts, while in the dark they did not. In the sunlight, nearby these green parts, small bubbles are being produced. And in absence of light, these bubbles were not seen. Later, he identified this bubble to be oxygen. And later on, he did the analysis. He found that these bubbles are nothing but these are oxygen. Thus, he showed that only the green parts of the plant release oxygen. So these were some of the experiments related with this chapter. Then there was one more experiment by Julius von Sack. He have conducted his experiment in the year 1854. He proved that glucose is produced when plant grow and it is usually stored as starch. Chlorophyll is located in special bodies that is called chloroplasts. Glucose is made in the green parts of the plant. So he said that during photosynthesis, sunlight is required, carbon dioxide is required, water is required, and what are the byproduct? What are the product? Oxygen are released in the environment, and glucose is stored in the plant body and consumed for performing various activities. Then there was experiment that was done by T.W. Engelmann. What he did, he split the light using a prism. This is a light. He have passed this light through prism, and then he have done, he split the light using a prism into a special component and illuminated a green alga cladophora. This is what green alga cladophora. Placed in a suspension of aerobic bacteria. The bacteria were used to detect the right sides of oxygen evolution. So wavelength, Vibgyor, you may be knowing Vibgyor. So the wavelength, range of wavelength from Vibgyor to 400 to 700 nanometer. So aerobic bacteria, he have placed cladophora. He observed that the bacterium accumulated mainly in the region of blue and red light. This is a bacterium. Their population, they get accumulated in this region, blue region and red region of the spectrum. It was the first described action spectrum of the photosynthesis. It resembled the absorption spectrum of chlorophyll A and B. So what he said that the maximum photosynthesis takes place in blue and red region. More oxygen is released. So if more oxygen is released, means more photosynthesis takes place in what? Blue and red color of the visible spectrum. By the middle of 19th century, it is discovered that plants use light energy to make carbohydrate from carbon dioxide and H2O, water. Empirical equation of the process of photosynthesis is carbon dioxide plus H2O. In presence of sunlight, they produce starch and release oxygen, where CH2O represents a carbohydrate, that is what? A glucose. So this is what basic formula for empirical equation of the process of photosynthesis. So they take what? Carbon dioxide from the environment, water from the soil, and in presence of sunlight, they perform what? They really produce what? Starch, and they release carbon dioxide. Experiment done by Cornelius van Neel. Van Neel, he was a microbiologist. Microbiologist, the person who study about microorganism, they are known as microbiologist. So Van Neel, he was a microbiologist, conducted some studies in purple and green bacteria. This is about purple bacteria and green bacteria. He demonstrated that photosynthesis is a light-dependent reaction in which hydrogen from the suitable oxidizable compound reduces carbon dioxide to carbohydrate. What happened? It is what light-dependent reaction. First, it is what light-dependent reaction in which hydrogen from suitable oxidizable compound. Here, suitable oxidizable compounds in case of this normal plant, that is what? Water. And then oxidizable compound reduces carbon dioxide to carbohydrate. This can be expressed as by H2A. This is what? Suitable oxidizable compound. Then it can convert carbon dioxide into what? Starch, release water, and then also so here In plant, S2 is the hydrogen donor. In plant, S2 is the hydrogen donor and oxidized to oxygen. S2O is what? Water is the hydrogen donor and oxidized to oxygen. So here the oxygen which is released here, it is coming from carbon dioxide, yeah, water. So it is coming from it is coming from water, not from carbon dioxide. Purple and green sulfur bacteria use hydrogen sulfide as a hydrogen donor. So the oxidation product is sulfur or sulfate and no oxygen is produced. Sulfur bacteria, sulfur will be oxygen. Sulfur will be produced and oxygen will not be produced. Thus, he inferred that the oxygen evolved by green plant come from S2O, not from carbon dioxide. Sometime question will be there. During the process of photosynthesis, both carbon dioxide and S2O is used. So the oxygen which is evolved, it comes from carbon dioxide or water. So it comes from water, not from carbon dioxide. This was later proved by using radioisotope techniques. Therefore, overall correct equation for photosynthesis is CO2 plus 12OH2 in presence of sunlight converted into C6H12O6, 6H2 plus 6O2. Now, photosynthesis, site and pigment. Where photosynthesis plays. So already you may have learned about the structure of chloroplast. So it is what cellular organelle present in plant cell, not in animal cell. So it is having outer membrane, having inner membrane. There are thylakoids are there, piles of grana that is called thylakoids. Here and this space is called stroma. Here there is stroma lamellae. So outer membrane, inner membrane, this is called grana. Grana is nothing. What is it? It is when many thylakoids are kept piled up, that is called grana. This is what grana. Then this is stroma. There is ribosomes are also there. Starch granules are there. Lipid droplets are also present in chloroplast. So photosynthesis takes place in green leaves and other green part of the plant. Chloroplasts present in the wall of mesophyll cells of leaf. There are special type of cells present in the leaves. They are called mesophyll cells. In mesophyll cells, there is a chloroplast is present in the wall of mesophyll cells. It helps to get optimum quantity of incident light. Chloroplasts contain a membranous system. It consists of grana, stroma, lamellae, and fluid stroma. Each grana is a group of membrane-bound sacs called thylakoid. Thylakoid, other name lamellae. They contain leaf pigments. So leaf pigments are present in these thylakoids. The membrane system trap light energy. The membrane system, there is a membrane system trap the light energy and synthesize ATP and NADPH. NADPH is ATP stand for adenosine triphosphate and NADPH stand for reduced phosphorylated nicotine adenine dinucleotide. So first we study we can say like this: nicotine, adenine dinucleotide, phosphorylated and reduced. So yeah, it is better to say reduced phosphorylated NAD. NAD stand for nicotine adenine dinucleotide. It is called light reaction. So membrane system, there is what? Production of ATP and NADPH. It is called light reaction. So light reaction, what happened in light reaction? There is a production of ATP and NADP. What is involved here? Water is involved, and water is gives hydrogen, and then hydrogen reduce this carbon dioxide into glucose, a carbohydrate. And this takes place in, for this, light is not required. So in this way, oxygen is released. So sometime question will be there, what are the product of light reaction? So answer will be ATP and NADPH as well as oxygen. What is the product of dark reaction? Glucose. In stroma, enzymatic reaction incorporate carbon dioxide into plant for synthesizing sugar, which in turn form starch. It is called dark reaction. It does not mean that they occur in darkness or they are not light dependent. So if light is there, only then ATP will be produced. When it is only then this reaction will be there. So it is, we cannot say that it is not dependent on light. Yeah, it doesn't take place in the presence of light. It will take place. Pigment involved in photosynthesis. Now we'll be discussing about pigment involved in photosynthesis. Pigments are substances that have ability to absorb light at specific wavelength. So we have seen the leaves, they are green. We can say that they are green, but they are not green. There are different shades of green. So there is one technology that is called chromatography. Shows the following leaf pigment in chromatograph. Chlorophyll A, bright or blue-green. Chlorophyll A, because of chlorophyll A, the color of leaves is bright or blue-green. Chlorophyll B, yellow-green. Chlorophyll A, that is called bright or blue-green. Chlorophyll B, yellow-green. Xanthophyll, yellow. And yellow to yellow-orange, carotene. So these are the pigments. These can be separated by using one technology that is called chromatography. So here, this is a chromatography of different chlorophyll. So chlorophyll A is what? Bright and blue-green. You have to learn all these things. Learn and memorize. Chlorophyll A, bright or blue-green. B, yellow or green. Xanthophyll, yellow, and carotene, yellow or to yellow-orange. Chlorophyll A is the main. Chlorophyll B, xanthophyll, and carotenoid, they are accessory pigments. So sometime the question will be there, which one is the main pigment? So answer will be what? A. What are they? Chlorophyll B, xanthophyll, and carotenoids. So they are accessory pigments. Now, function of accessory pigments: they absorb light at different wavelength and transfer the energy to chlorophyll A. They protect chlorophyll A from photooxidation. Now, pigment involved in photosynthesis. The absorption spectrum and action spectrum coincide closely, showing that photosynthesis is maximum at blue and red region. See, this is what blue region, this is the red region, blue and red. So there is a maximum absorption of light in this region as well as this region. So maximum absorption, maximum photosynthesis. Here, light absorb maximum in blue and red, and rate of photosynthesis maximum in blue region and red region. So this absorption spectrum and action spectrum, they are what? Overlapping. This is what? Absorption spectrum, and this is what? Action spectrum. When absorption spectrum is more, action is all, rate of photosynthesis is also more. When absorption spectrum is less, photosynthesis is also less. So they are what? Overlapping. Sometimes question will be there, photosynthesis is maximum in which region of the spectrum? So you have to answer that blue and red region of the spectrum. The graphs also shows that chlorophyll A is the chief pigment. Chlorophyll A is what? Chief pigment associated with photosynthesis. Here also chlorophyll A is synthesizing, absorbing more light, and here also. Here, so chlorophyll A is the chief pigment. So this graph showing what? Wavelength and absorption of light by chloroplast pigments. So graph showing absorption spectrum of chlorophyll A, B, and carotenoid. This is chlorophyll A, this is a B, and this is carotenoid. This is graph showing an action spectrum of photosynthesis. Photosynthesis is maximum in what? A blue and red region. This graph showing action spectrum superimposed on absorption spectrum of chlorophyll A. So there is more photosynthesis in the A region. This is A here and here. So here and here there is a absorption is maximum as rate of photosynthesis is also maximum. Therefore, chlorophyll A is the chief pigment associated with photosynthesis. Now, photosystems. We'll be discussing about photosystem. Photosystem. Pigments are organized into two photosystems called photosystem first, PS1, and photosystem second, that is called PS2. These are named in the sequence of their discovery. Each photosystem has a chlorophyll A and accessory pigment bound by protein. So here this is your chlorophyll A, and there are accessory pigments bound by all these things. So this is what, there is one complex, light harvesting complex, yeah, LHC. Sometimes question will be there, what do you mean by LHC? So LHC stand for light harvesting complex. The sunlight is nothing. They are made up of photon. Photon are absorbed by chlorophylls. There are some reaction sector. So if there in photon means energy, so here this molecule they are getting energy. The electron will get excited. Then there will be some electron acceptors are there. They are called primary acceptors. All pigments except one molecule of chlorophyll A form a light harvesting complex, LHC or antenna. All pigment except one molecule of chlorophyll A, only one molecule of chlorophyll A, it will be what? Will remain isolated, and others will form a light harvesting complex. Single chlorophyll A act as a reaction center. So which chlorophyll work as a reaction center? Chlorophyll A. One molecule of chlorophyll A will be working as a reaction center. In photosystem first, in photosystem first, the reaction center absorbs light at 700 nanometer and so-called P700. So photosystem first is also known as P700. Why? Because the reaction center absorbs light at 700 nanometer. In photosystem second, the reaction center absorbs light at 600 light of wavelength 680 nanometer and called P600. So आपको लर्न करना है, जिसका मार्क ज्यादा है तो फर्स्ट आएगा, जिसका मार्क कम है तो सेकेंड आएगा. तो फोटोसिस्टम फर्स्ट 700, photosystem second 680. Light reaction are photochemical phase. Now we'll be learning light reaction are photochemical phase. If we see this diagram, light is absorbed by this plant, then there is a, this is a photosystems are there. This is a photosystem second is there. Photosystem second is having what? Reaction center is what? 680. So light through light, this molecule will get what? Photon. So one molecule, chlorophyll molecule of that is P608, it will become a reaction center. Now here there will be splitting of water molecule. This 2s2 in the presence of MnO4 Mn4O. This for splitting of water, manganese is mineral which is required. So sometimes the question is also is there na which minerals facilitate splitting of water molecule? So that answer will be what? Manganese. So this water will be splitting and it will be releasing oxygen as well as hydrogen ion. Hydrogen ion means proton. Then this photosystem, the electron will get excited, and this excited electron will be picked by electron acceptors. So this, then this will be photosystem second is connected with photosystem first through this electron transport system. This is electron transport system. There are many components here in electron transport system. We'll be learning it in what? Respiration chapter. So there are many components like cytochrome B is there, A1 is there, cytochrome. So many cytochromes are there. Plastoquinone is there. So these electrons, that is called, they will be picked up by this photosystem, and they will be, then this ADP will be converted into ATP. And this electron will be transferred from photosystem second to photosystem first. Now this will also get excited with this. Here this reaction center is P700. So it will be getting electron from photosystem second, as well as it will be getting energy photon from this sunlight. It will get excited. Then there is what? Excited electron receptors are there. Then here what will happen? NADP will be reduced into NADPH. So here NADP is nothing but it is energy rich compound. 1 NAD is equal to 3 ATP. Light reaction, that is also known as photochemical phase. So we'll be discussing about light reaction. It includes light absorption, water splitting, oxygen release, formation of ATP and NADPH. So what are different components of light reaction? First is light absorption, then water splitting. Light absorption is first. This is light will be absorbed. Then water splitting. Water will split. This water molecule will split. It will split and it will release what? Hydrogen ion, that proton, and it will release what? Oxygen. Oxygen will be released and proton will be used to oxidize, to reduce carbon dioxide into what? Sugar. So water is splitting, oxygen is released, formation of ATP. What will be produced? ATP, adenosine triphosphate, NADPH, nicotine adenine dinucleotide phosphorylated and reduced. So these are the product of light reaction. This cycle is called Calvin cycle, and ultimately sugar is also produced. Sugar is not produced during the light reaction. It will produce, this material will be used, product of light reaction will be used during sugar production. So ATP and NADP, what are ATP? You already may be knowing what is ATP. What do you mean by NADPH, they are nothing, high energy chemical intermediates. Then electron transport, Z scheme. It is what sometimes it is known as, popularly known as Z scheme. So we can say like Z. This is like Z. Yeah, Z scheme. When photosystem second, this is a photosystem second, absorbs red light of 680 nanometer. It will absorb the sunlight of which wavelength? 680 nanometer. This is also known as red light. Red light means the sunlight with the wavelength of 680 nanometer, that is called red light. So photosystem second absorb which light? Red light. Red light, what is the wavelength of red light? 680 wavelength. Electrons are excited and transferred to an electron acceptor. So here electron will get excited and they will be accepted by electron acceptors. The electron acceptor passes them to a chain of electron transport system. So, and again, this electron will be transported to one system that is called electron transport system. This is what electron transport system. This electron from here will go here. From here, it will go to electron acceptor. From there, it will go to electron transport system, consisting of cytochrome. Here, electron transport system is made up of cytochromes. What are the component of electron transport system? It is consists of cytochromes. This movement of electron is downhill in terms of redox potential. So it is what? Downhill. The electrons are transformed. This is what? Uphill, and here this is downhill. The electrons are transferred to the pigment of photosystem first. So from photosystem second, electrons are transferred to photosystem first. And then from here also, this area will be also this light harvesting complex of photosystem second. It will also get light and it will also get electrons. So here reaction center is what? 700. Wavelength is what? 700. So here again electron acceptor, and this electron acceptor will be reducing NADP ion into NADPH. Simultaneously, electron in photosystem first are also excited when they receive red light of 700 nanometer and are transferred to another receptor molecule having a greater redox potential. So this is having less great redox potential. Photosystem first, second is having less redox potential in comparison to photosystem first. So less redox potential, so less ADP will be produced here, more powerful. So NAD is more powerful than ATP. These electrons are moved downhill to molecule of NADP ion. As a result, NADP ion is reduced to NADPH and protons are released. Proton means hydrogen ions are released. Transfer of electrons from photosystem second to photosystem first and finally downhill to NADP ion is called Z scheme due to its zigzag shape. Zigzag shape. Yeah, Z. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. So आगे बढ़ते हैं. Let us now splitting of water, that is called photolysis. So what is splitting of water? The water splitting complex in photosystem second. Water splitting component is present in which photosystem? Photosystem second. It is not present in photosystem first. So sometime question will be there. Water splitting complex is present and photo related with photosystem second or first. So you have to mention it is what? Related with photosystem second. On the inner side of thylakoid membrane. They all are located on the inner side of thylakoid membrane, not outer side. Water is split into hydrogen ion and oxygen and electron. What are the product of water splitting? Hydrogen ion, oxygen and electrons. So these electrons are getting excited. These electrons are coming from splitting of water molecules. So photosystem second can supply electron continuously by replacing electron from water splitting. Thus, photosystem second provide electron needed to replace those removed from photosystem first. Here there is no any source of, in photosystem first, there is no any source of electron. So source of electron is what? Photosystem second. The proton, these proton which are released here, are used to reduce NADP to NADPH. These protons, they are used to reduce what? NADP to NADPH. Oxygen is liberated as byproduct of photosynthesis. So oxygen will be liberated as a byproduct of photosynthesis. This is all about splitting of water, that is also known as photolysis. Sometimes question will be there. What do you mean by photolysis? What are the product of photolysis? So proton, oxygen and electron, they are product of splitting of water molecule. It is a part of photosystem second. Now let us proceed. Now photophosphorylation. Photophosphorylation. Okay. Synthesis of ATP by cell in mitochondria and chloroplasts. So synthesis of ATP, where it is synthesized? In mitochondria or chloroplast is called phosphorylation. What is phosphorylation? Because this is what? Aided by light, facilitated by light, therefore it is called what? Photophosphorylation. So synthesis of ATP by mitochondria or chloroplast is called phosphorylation. Phosphorylation is the synthesis of ATP from ADP in chloroplast in presence of light. It occurs in two ways, non-cyclic and cyclic. So there will be what? Non-cyclic photophosphorylation and there will be cyclic photophosphorylation. Now we'll be discussing what is non-cyclic photophosphorylation and what is cyclic photophosphorylation. Now non-cyclic photophosphorylation. You already know phosphorylation means synthesis of ATP from ADP either in chloroplast or mitochondria, that is called phosphorylation. Then there is what is non-cyclic photophosphorylation. It occurs when the two photosystem work in a series. First photosystem second and then photosystem first through an electron transport chain as seen in Z scheme. Here ATP and NADP plus hydrogen ion are synthesized. In non-cyclic, what are produced? ATP and NADP plus hydrogen are synthesized. It is a non-cyclic process because the electron lost by photosystem second do not come back to but pass on to NADP. So Z scheme is what? Non-cyclic photophosphorylation. So because here electrons are going from here, they are not coming back, but they are going and then they are from photosystem second, then put to photosystem first. So this is what? Non-cyclic photophosphorylation. Now cyclic photophosphorylation. It occurs in stoma, lamellae, where only photosystem first is functional. Only when photosystem first is functional, then it occurs in stoma lamellae. The electron is circulated within photosystem. Here, in photosystem first, there is a light. Then there is a getting electron from light as well as this through electron transport system. Then here electron acceptor is there. There is ATP. ADP is converted into ATP. So there is no production of NADP. Only ATP is synthesized. The electron is circulated within photosystem and ATP synthesis occurs due to cyclic flow of electron. The lamella of the grana have photosystem first and photosystem second. The stoma lamella membrane lack photosystem second. And NADP reductase. The electron does not pass to NADP ion but is cycled back to photosystem first through electron transport chain. Here only ATP is synthesized, no NADP and hydrogen ion. Cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nanometers are available. So this is what cyclic photophosphorylation. Now, chemiosmotic hypothesis. What is chemiosmotic hypothesis? Something related with osmosis, higher concentration to lower concentration. So it explains mechanism of ATP synthesis in chloroplasts. So what does chemiosmotic hypothesis explains? It explains mechanism of ATP synthesis in chloroplasts. Now what is chemiosmosis? Movement of ion across a semi-permeable membrane. These are ion, this is a membrane. So movement of ion across semi-permeable membrane, that is called chemiosmosis. It occurs in chloroplast and mitochondria. Where you will find chemiosmosis? It occurs in chloroplast as well as mitochondria. So here, movement of ions across semi-permeable membrane, that is called chemiosmosis. Chemiosmosis need a membrane. What is required? It need a membrane, a proton pump, proton gradient across thylakoid membrane, and ATP synthase. This is what ATP synthase enzyme. Splitting of water molecule takes place in the inner side of membrane. This is a membrane, thylakoid membrane. There is a what? Outer side and inner side. So in inner side there is what? Inner side of this thylakoid membrane. This is what? Thylakoid membrane. Outer side and inner side. So inner side there is a splitting of water molecule. So what is the product of this splitting of water molecule? Hydrogen ion, oxygen. This is a product. Because of this, on this membrane, there is a development of what? Proton gradient. What develop? Sometimes the option proton gradient, electron gradient, ion gradient. So you have to mention what? Proton gradient. Always ensure that you are writing what? Proton gradient. Proton gradient develop across thylakoid membrane, not stroma membrane. It is what? Across thylakoid membrane. And there is one more requirement, ATP synthase enzyme. This enzyme is what? ATP synthase. Splitting of water takes place on the inner side of the membrane. So the proton accumulate in the lumen of thylakoid. This is thylakoid, this is the lumen of thylakoid. So proton accumulate in the lumen of thylakoids. Then what happens? As electrons move through photosystem first, there is a photosystem second, and this is photosystem first. They are connected. Both of them are connected by electron transport system. So this is a part of electron transport system, which include plastoquinone, cytochromes, and plastocyanin. These are nothing but they are component of electron transport system. As electrons move through photosystem, protons are transported across membrane. So in stroma also there will be some proton will be there. They will be transported from outside, from stroma to lumen of thylakoid. It is due to the removal of proton from stroma for the following reason. The primary electron acceptor is located toward the outer side of membrane. The primary electron acceptor is located towards the outer side of membrane and transfer its electron to hydrogen carrier. So this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to electron carrier on the inner side of the membrane, proton is released into the lumen of the membrane. So here there are so many electron carriers are there. They are transporting electron from one area to another, from one cytochrome to another. When they are transporting electron, and they need hydrogen acceptor also, they are transporting hydrogen inside in the lumen. As electrons move through photosystem, protons are transported across membrane. It is due to the removal of proton from stroma for following reason. The NADP reducted enzyme is located on stroma side. So NADP reducted enzyme is located on stroma side of the membrane. Here, this is what NADP reducted enzyme is located this side, not this side. So NADP reducted enzyme is located on stroma side of membrane. Along with electron coming from photosystem first, protons are necessary to reduce NADP ion. These protons are also removed from the stroma. So some of the protons which are there in the stroma, from here also they are removed. Hence, proton in stroma are decreased. Ultimately, proton from here in stroma, they are used in production of NADP. Some of the proton which are there in stroma, they are accepted by electron acceptor. This cytochrome component of the electron transport system, and they are transporting this proton inside this lumen. Therefore, concentration of hydrogen ion or proton become very less in stroma, and it become high in what lumen. Because of this, it creates a proton gradient across the thylakoid membrane and there is a decrease in pH in lumen. So more hydrogen ion, more proton, less pH. So it decreases what pH of what lumen. Because of decreased breakdown of proton gradient lead to the synthesis of ATP by ATP synthase enzyme. Name the enzyme which is used to synthesize ATP. The answer will be what? ATP synthase. ATP synthase consists of two parts. This is ATP synthase. This is ATP synthase. It is made up of two parts. This is outside. This part is outside. This part is inside. This is the inside. So it is having two parts: CF0, CF1. CF0, it is embedded in the membrane and form a transmembrane channel. It is embedded, see here, it is embedded in the membrane. So it produces what? Transmembrane channel. It carries out facilitated diffusion of proton across the membrane to stroma. It results in breakdown of proton gradient. Second component of ATP synthase is CF1. It protrudes on the outer surface of thylakoid membrane. The energy due to breakdown of gradient causes a conformational change in the CF1 particle. It makes the enzyme to synthesize ATP. Then it will be about synthesizing ATP from ADP. Energy is used to pump proton across a membrane. This energy is used to pump proton across a membrane to create a gradient or a high concentration of proton within the thylakoid lumen. ATP synthase has a channel for the diffusion of proton back across the membrane. This releases energy to activate ATP synthase that catalyzes formation of ATP. Now we'll be discussing about photosynthesis, that part that is called dark reaction, or it is also known as what? Biosynthetic phase. Here, whatever is produced during what? Light reaction, that is ATP, NADP, they are being used up. Product of light reaction, that are ATP, NADPH, and oxygen. Dark reaction is the use of ATP and NADPH to drive the process for synthesis of food, that is what? Sugar. So this is light energy. There is whatever product are produced, they are utilized during dark reaction, and then they produce what? Organic molecule. This phase does not directly depend on light, but is dependent on the product of light reaction. So it is not dependent on light, but it is dependent on the product of light reaction. So it can be verified as follows. Immediately after light become unavailable, Biosynthetic process continue for some time and then stop. If light is available, the synthesis start again. So if as soon as light is available, there will be production of ATP. If ATP and NADP is there, the reaction will take place. No light. If there is no light, so for some time there will be what production of ATP and NADP. Then this drug reaction will continue. But after some time, no light, no production of ATP, no production of NADP, therefore no dark reaction. So indirectly dark reaction is dependent on not light, but dependent on product of light reaction. What happened here? Dark reaction, biosynthetic phase. Carbon dioxide combined with water to form CH2ON or sugar. Here carbon dioxide plus water. They form what? CH2 starch or sugar. CO2 carbon dioxide assimilation during photosynthesis is a two type. C3 pathway and C4 pathway. What is C3 pathway? In C3 pathway, the first product of carbon dioxide fixation is C3 organic acid. That is called 3-phosphoglyceric acid or PGA. So first product of C3 plant is what? 3 carbon containing compound, that is organic acid, that is called PGA. PG stands for phosphoglyceric acid. This was discovered by Melvin Calvin. Therefore, it is also known as Calvin cycle. There is another pathway, C4 pathway. In this first stable product is azelaic acid, OAA. It is a 4 carbon acid. Therefore, it is known as C4 pathway. So this is what C3 pathway, that is called Calvin cycle. And then there is a C4 pathway. So we will be discussing all these things in detail. Now C3 pathway. This pathway occurs in all synthetic plants, whether this is what C3 or C4. It has 3 stages: carboxylation, this is carboxylation, then reduction, then regeneration. So Calvin cycle is having 3 stages. First is what? Carboxylation, then reduction, then third is what? Regeneration. Now what happened in carboxylation? Here there is a carboxylation of RUBP. What is this? There is a carboxylation of RUBP. RUBP stands for ribulose biphosphate. It is a 5-carbon ketose sugar. It is what? 5-carbon ketose sugar. It is the primary carbon dioxide acceptor. So sometimes question will be there, RUBP, what is RUBP? Expand RUBP. So you have to expand it, right? Ribulose biphosphate. How many carbons are there in RUBP? It is having 5 carbons. Which type of sugar? It is a ketose sugar. What is the primary receptor of the carbon dioxide? RUBP. It is the most crucial step. Carbon dioxide is fixed by RUBP to 2 molecules of carbon dioxide is fixed by RUBP to 2,3-PGA in presence of enzyme RUBP carboxylase. There is 2 molecules of PGA are produced. Okay. And there is one enzyme. This is enzyme related process that is called what? RUBP carboxylase. Since this enzyme also has an oxygenation activity, therefore it is called Rubisco. Rubisco stands for ribulose biphosphate carboxylase and oxygenase, because this enzyme is having affinity for both oxygen as well as carbon dioxide as well as oxygen. RUBP, Rubisco, is most abundant enzyme in the world. Sometimes question will be there. Name which among following is the most abundant enzyme in the world. So answer will be what? Rubisco. So this is what? Atmospheric oxygen and water in presence of this ketose sugar RUBP. RUBP undergo carboxylation, produces 2 molecules of 3-PGA. 3-PGA. PGA stands for phosphoglyceric acid. Then there is a reduction. ATP is consumed, NADP is consumed, then it undergo 3-PGA undergo reduction, then it produces triose phosphate. This triose phosphate ultimately converted into sucrose and starch. Again, there will be what? Regeneration. Then here ATP molecule will be converted into ADP. Then RUBP will get what? Regenerated. So this is what all about C3 pathway. Practice it and learn it. Next, carboxylation. And now second stage of this C3 is dark reaction is what? Reduction. It is a series of reaction leading to the glucose formation. So now, in this way, what happened in reduction? It is a series of reaction leading to glucose formation. Here, two ATP molecule for phosphorylation and two NADPH for reduction per carbon dioxide molecules are used. Fixation of 6 carbon dioxide molecules and 6 turns of cycles are needed to remove one glucose molecule from pathway. So to release one glucose molecule from the Calvin cycle, this cycle how many turns are required? 6 turns are required to produce one molecule of glucose from C3 Calvin cycle. How many turns of the cycles are needed? 6 turns are needed. So 1 turn, 2 turn, 3 turn, 4 turn, 5 turn, 6 turns are needed. Now third is what? Regeneration of, third phase of Calvin cycle is what? Regeneration of RUBP. It is crucial for continuation of the cycle. It requires one ATP for phosphorylation to form RUBP. How many ATP are required? One ATP is required. Hence, for every carbon dioxide molecule, 3 ATP molecules and 2 NADP are required. So it is very there is a great requirement to fix one molecule of carbon dioxide. How many ATP molecules? 3 ATP molecule and 2 NADP molecules are required. It is probably to meet this difference in number of ATP and NADP used in the dark reaction that the cyclic phosphorylation takes place. To make one glucose molecule, 6 turns of the cycle are needed. Now, C3 pathway Calvin cycle, what goes in and what comes out of Calvin cycle? How many molecules of carbon dioxide? 6 molecules. And how many, what is out? 1 glucose. 18 molecule of ATP. 2 here, 1 here. 6. To fix one carbon molecule, 6 ATP are required. So to complete one glucose molecules, 6 3s are 3 into 6. 3 into 6. Here 3 into 6. Why 3? Why 3? To fix one molecule of carbon dioxide, 3 ATP are required. 2 here and 1 here. So to complete produce one molecule of sucrose, 6 times it should, this cycle should turn 6 times. So 6 3s are 8, 18 ATP are required. Here in one turn, how many are required? 2 are required. So how many to produce one molecule of glucose? 6 are required. So it becomes what? 12 NADP. So to fix one molecule of, to produce one molecule of glucose, 18 ATP, 12 NADP are required. Now there is another pathway that is called C4 pathway. This is also known as Hatch and Slag pathway. Hatch and Slag pathway. It is present in plant adapted to dry tropical region. This is Earth. Earth is having some region nearby equator that is called tropical region. They also use C3 pathway as main biosynthetic pathway. C4 plant, there is both C3 pathway as well as C4 pathway, but in C3 plant only C3 cycle is there. If we see the leaves anatomy of the plant here, this is what? Vascular bundle. Vascular bundles are covered by bundle sheath. And here bundle sheath are having chloroplast. So there are the mesophyll cells, and then there are what? Bundle sheath cells are there. Bundle sheath cells are having what? Chloroplast. So this is anatomy of this C4 plant. So they shows what? They shows what? Cran's anatomy. What is Cran's anatomy? Where you will find Cran's anatomy in C4 plant. The large cell around the vascular bundle of the C4 plant are called bundle sheath cell. Such anatomy is called Cran's anatomy. The bundle sheath cells may form several layers around the vascular bundle. Not only one layer, they may form what? Several layers. They have large number of chloroplasts, thick wall, impervious to gas exchange, and no intercellular space. Now steps of Hatch and Slag pathway. Here, primary carbon acceptor is phosphophenyl pyruvate, PEP. Here, phosphophenyl pyruvate is the acceptor of carbon dioxide. Phosphophenyl pyruvate is that is called PEP. It is what? 3 carbon molecule. RUBP is what? 5 carbon molecule. In C3, the primary acceptor is RUBP, and here it is what? PEP. PEP stands for phosphophenyl pyruvate. It has 3 carbon molecules seen in mesophyll cells. They are present in what? Mesophyll cell. The enzyme for this fixation is PEP carboxylase. There is one enzyme here that will be called what? PEP carboxylase. It is also known as pepcase. The mesophyll cells lack Rubisco enzyme. In mesophyll cells, there is no Rubisco. The C4 acid OA is formed in the mesophyll cell. So here C4 acid Rubisco is not there here in mesophyll cell. Instead of Rubisco, what is there? Peptase is there. So here C4 acid, that is called oxaloacetic acid, is produced, is formed in the mesophyll cell. It then forms other 4 carbon acid like malic acid as aspartic acid. Apart from this C4, it also produces C4 acid, that may be what? Malic acid or aspartic acid. They are transported to bundle sheath cell. So malic acid, aspartic acid, not OA. Malic acid, aspartic acid will be transported from mesophyll cell to bundle sheath cell. They are transported. Then in the bundle sheath cell, C4 acid are broken down to release carbon dioxide and C3 molecule is produced. In bundle sheath cell, this C4 acid will undergo decarboxylation, will release carbon dioxide, and C3 acid will be produced. Now this carbon dioxide will be fixed by Calvin cycle. So in bundle sheath cell, there will be Calvin cycle, and in here mesophyll cell, there will be Hatch and Slag path cycle in C4 plant. The C3 molecule is transported back to mesophyll where it is converted to PEP again. This C3 again transported to mesophyll cell where it will be converted to C3 acid. Then it will degenerate what? Phosphophenyl pyruvate. Bundle sheath cells are rich in Rubisco. Here, this cell, they are rich in what? Rubisco, but lack peptase. Thus, C3 pathway is common to C3 and C4 plant. Rubisco is here, but not here. Rubisco is present here in bundle sheath cell, but here Rubisco is not present. But so here in the bundle sheath cell, there will be C3 cycle. In mesophyll cell, there will be what? C4 cycle. C4 plants are special. Why they are special? Because they have a special type of leaf anatomy that is called Cran's anatomy. They tolerate higher temperature. They show a response to high light intensity. They lack photorespiration. There is no photorespiration. They have greater productivity of biomass. If we compare with C3, the C4 are having greater productivity of biomass. Why? Because in C4s are having greater productivity in comparison with C3. But why? Because in C4 there is no photorespiration, but in C3 there is a photorespiration. Now, what is photorespiration? In Calvin pathway, RUBP combined with carbon dioxide, RUBP combined with carbon dioxide in presence of Rubisco, it produces 2 molecules of 3-PGA. Here, active site of Rubisco can bind to carbon dioxide and oxygen, hence the name. Rubisco have what? Both binding site for carbon dioxide as well as oxygen. Therefore, it is known as Rubisco. Now, Rubisco has a greater affinity for carbon dioxide than for oxygen. This binding is competitive. Relative concentration of oxygen and carbon dioxide determine which will bind to the enzyme. So, this is Rubisco. It can bind with what? Oxygen as well as carbon dioxide. Now there is a competition between what? Carbon dioxide and oxygen. In the chloroplast, when this Rubisco is forming bond with what? Carbon dioxide, it will produce phosphoglycolate. In peroxisome and mitochondria, it will what? it will use oxygen and it will release carbon dioxide. So here, in C3 plants, some oxygen bind to rubisco. In C3 plants, some oxygen bind to rubisco. This is rubisco, some oxygen bind with rubisco. So there is what ribulose 1,5 biphosphate is there, RUBP is there. So RUBP will undergo carboxylation. It will produce what? 3-phosphoglycerate. But here, if oxygen is binding, chloroplast also, instead of producing PGA, it will produce what? 2-phosphoglycolate. It will produce what? 2-phosphoglycolate. Instead of producing what? 3-phosphoglycerate. In C3 plants, some oxygen bind to rubisco, hence carbon dioxide fixation is decreased. Here RUBP bind with oxygen to form one molecule of phosphoglycerate and phosphoglycolate. RUP when bind with oxygen to form one molecule of phosphoglycerate and phosphoglycolate. This pathway is called photorespiration. In this pathway, there is no synthesis of sugar, ATP and NADP. Photorespiration, there is no synthesis of sugar, ATP and NADP. Hence, photorespiration is a wasteful process. Rather, it causes the release of carbon dioxide by using ATP. In C4 plant, photorespiration does not occur because they can increase carbon dioxide concentration at the enzyme site. Therefore, rubisco will never bind with what? Oxygen. This takes place when carbon C4 acid formed from the mesophyll is broken down in the bundle cell to release carbon dioxide. This minimizes the oxygenase activity of rubisco. So oxygenase activity will not be there. Due to lack of photorespiration, productivity and yield are better in C4 plant. In addition, this plant shows tolerance to higher temperatures. C4 plant, they show what? They are tolerant to higher temperature. Now comparison between C3 and C4 plant. Here, photosynthesis occur in mesophyll cell in C3 and in C4, both mesophyll and bundle sheath cell. Cranch anatomy is absent in C3. Cranch anatomy is present in C4. RUBP is primary carbon dioxide acceptor in C3. Phosphophenol pyruvate is primary carbon dioxide acceptor in C4. 3-PGA, a 3-carbon compound, is first stable compound. Here in C4, OAA, oxaloacetic acid, a 4-carbon containing compound, is first stable product. In C3, chloroplasts are only one type. They are present in granule. Here in C4, chloroplasts are dimorphic. They are present in granule in the mesophyll and agranule in bundle sheath. In C3, photorespiration loss is high. Photorespiration is absent or negligible in C4. In C3, high carbon dioxide compensation point, 25 to 100 ml is required. Here in C4, low carbon dioxide compensation point is required. Optimum temperature for photosynthesis is about 25 degrees Celsius in C3. In C4, it is about 35 degrees to 45 degrees Celsius. Photosynthetically less efficient and productivity is low. Rice, wheat, bean, potato, they are example of C3 plant. Maize, sugar cane, amartham, sorghum, they are the example of C4 plant. So you remember all these points, example as well. Now we'll be discussing about factors affecting photosynthesis. So there are two factors which will be affecting photosynthesis: internal, that is related with plant factor, and external. So internal factor include number, size, age, and orientation of leaves, mesophyll cells, and chloroplast. And internal carbon dioxide concentration, amount of chlorophyll. External factor includes sunlight, temperature, carbon dioxide concentration, water. Plant factors are dependent on genetic predisposition and growth of the plant. So plant factor are related with their genetic makeup. Then there is one law, that is Blackman's law of limiting factor. It was given by Blackman in the year 1905. If a biochemical process is affected by more than one factor, its rate is determined by the factor nearest to its minimal value. When there are so many factors, they are affecting a biochemical process. So the rate is determined by the factor nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. So there is about photosynthesis many factors. Light is there, carbon dioxide is required, oxygen is there, water is required. Okay, if light is there, carbon dioxide is there, no water, can photosynthesis take place? No. So in that case, water will become limiting factor. Simple. Example, a plant with green leaf, optimal light and carbon dioxide may not photosynthesize in very low temperature. If optimal temperature is given, it will start photosynthesis. So suppose there is a plant is there, plant is having green leaf, they are having optimum light, carbon dioxide is there. Even in if they is all these things are there, they will not perform photosynthesis if the temperature is very low. So temperature will become a limiting factor. So if you increase the temperature, it will start synthesizing performing photosynthesis. Now light as a limiting factor. Light quality, light intensity, and duration of exposure to light influence photosynthesis. There is a linear relationship between incident light and carbon dioxide fixation at low intensity of light. If the light of intensity is less, here it is what less, this area less. You increase the intensity of light, there will be increase in rate of photosynthesis. There is linear relation from here point 0 to A, and then at higher light intensity, the rate does not further increase. If you keep on increasing the intensity, photosynthesis will not increase. It will not go like this, because other factor become limiting. Light saturation occur at 10% of the full sunlight. Hence, except for plant in shade or in dense forest, light is rarely a limiting factor. Light saturation occur at 10% of full sunlight. So if only 10% of sunlight is available, that is sufficient for photosynthesis. High increase in incident light break down the chlorophyll, it decreases photosynthesis. If the intensity of light is very high, then it will damage the chlorophyll, and if there is a chlorophyll is damaged, it will decrease photosynthesis. Now carbon dioxide concentration, which it will be also affecting rate of photosynthesis. Major limiting factor for photosynthesis is carbon dioxide. Carbon dioxide concentration is very low in the atmosphere, 0.03 to 0.04%. Increase up to 0.5% causes increase in carbon dioxide fixation rate. Beyond this level, it becomes damaging over longer period. So this is what optimum concentration of carbon dioxide. At low light conditions, C3 and C4 plant do not respond to high carbon dioxide condition. At high light intensity, they show increased rate of photosynthesis. C4 plants show saturation at about 360 ml per liter. C4 plant respond to increased carbon dioxide concentration and saturation is seen only beyond 450. Thus, current availability of carbon dioxide level is limiting to the C3 plant. Due to respond to higher carbon dioxide concentration, C3 plant shows increased photosynthesis and high productivity. This fact is used for some greenhouse crop like tomato and bell pepper. So they are grown in greenhouses because in greenhouses the concentration of carbon dioxide is more, so the perform more rate of photosynthesis will be more. They are grown in carbon dioxide enriched atmosphere, that is to higher yields. Now dark reaction. Temperature is another factor which affect photosynthesis. Dark reaction being enzymatic are temperature control. Influence of temperature on light reaction is very less. C4 plant respond to higher temperature and so higher rate of photosynthesis. More temperature, more photosynthesis in C4 plant. C3 plant have a much lower temperature optimum. The temperature optimum of plant also depend on their habitat. Tropical plant have a higher temperature optimum than the plant adapted to temperate climate. Water is another factor which affect the rate of photosynthesis. Water stress means shortage of water, closes the stomata, hence reduce the carbon dioxide availability. Water stress also wilt leaves. There is a water problem, so leaves are getting wilted. Thus reduce the surface area of the leaves and their metabolic activity. So thank you this much for this. This much is there in the syllabus. Now I will be signing up. Thank you very much. If you have not subscribed my channel, subscribe my channel to get many more videos related with class 11th and class 12th biology.